PDGF signaling with Kacey and Leilah. How is the body able to repair itself after an injury? This is through the platelet-derived growth factor, or PDGF, which is a key mechanism for cellular communication, particularly for wound healing. The PDGF molecule is hydrophobic, meaning it cannot pass through the cell membrane on its own. Instead, it needs receptor proteins on the cell's surface to transmit its signals. The group of receptor proteins it belongs to are called RTKs and are primarily used to regulate fundamental cellular processes like cell growth and division, which are also called proliferation. Their purpose is to link an external signal to a specific cellular response within the cell. One major advantage of RTK signaling is its high specificity and efficiency, which allows for strong signal amplification and a targeted response, crucial for wound healing as well as embryonic development. However, one major disadvantage is that when the receptors are overactive or mutated, they can trigger uncontrolled cell growth, which is a common cause of cancer.
One of the main components is the initial signaling molecule or ligand PDGF. This is the protein released by cells, such as platelets at the site of the wound. Platelets are tiny blood cell fragments that make blood clots to stop bleeding. This marks the beginning of the repair process. Another component, the PDGF receptor, is a protein that is embedded in the cell's membrane and acts as the receiver for the PDGF signal. It becomes active when the PDGF ligand attaches. Then, intracellular kinase proteins act as the cascade, passing and amplifying the signal from the receptor, ensuring the message reaches the cell's control center. The transcription factor is the final protein in the relay. It moves into the cell's nucleus and changes which genes are active, dictating the cell's response.
The process begins when a wound occurs, and the repair process starts immediately. Platelets at the site of the injury release the initial signaling protein, PDGF. The PDGF signal travels to a nearby target cell, where it is received by the PDGF receptor, embedded in the cell's membrane. This binding causes the receptor to change its shape, joining its two halves together and activating an enzyme. This enzyme then adds phosphate groups to the receptor. This activated receptor initiates a cascade of events inside the cell. It turns on the first of a series of intracellular proteins. Each protein in the relay activates the next one in the sequence, passing and amplifying the signal as it moves forward. The final protein in the relay is a transcription factor. It enters the cell's nucleus, where it binds to specific sites on the DNA and activates certain genes. With these genes now active, the cell produces the new proteins necessary for it to grow and divide. This is the intended cellular response: to repair damaged tissue and close the wound.
One example of an antagonist for PDGF signaling pathways is the drug Gleevec. Gleevec works as an oral cancer therapy that is able to inhibit the tyrosine kinase receptors, some of which are the pathways in PDGF. The drug fits into the receptors and stops them from being activated, preventing any downstream signaling and stops the molecules from changing shape. This is used with types of cancers such as leukemia because it can prevent the overgrowth of tumors in certain organs. By blocking these signals, Gleevec can prevent cell growth and division, which is the main cause of cancer.